Today, 3.2, enzyme catalysis. Now you see this word, we kind of touched on it the last time we talked about enzymes being catalysts or little protein molecules that speed up chemical reaction. So whenever you see catalysis, it just means causing an acceleration. Now go back and do some review. You know, enzymes, remember they are protein molecules. They're just structures made of protein, and their shape is extremely, has to be extremely precise, or their function doesn't happen in the way it should. There's 75,000 enzymes and proteins that we're discussing here. And then remember, they catalyze because they're catalysts. They speed up chemical reactions. Now, if you look at the picture over here, we use the green here, that's going to be the enzyme and the substrate. And if you go back, remember the substrate is what the enzyme is either breaking down or attaching it to something else. And it's using a chemical reaction to do so. Happens extremely quickly. And then this site here, it has to match because only one substrate will work with one enzyme, so they have to fit in. Now this is where the shape comes in, because if the enzyme shape is off, this might open up or close up, and then the substrate can't fit. Now keep in mind, while it's very specific, this enzyme and this substrate, they're like a lock and key. When this chemical reaction takes place and either breaks this up or puts this together into another molecule, this enzyme is free to keep on working, and it will work until it's worn out. And so it's very, very specific. The enzyme is to the substrate. So one enzyme, one substrate. Again, if you look at this, here's the substrate. It says hydrogen peroxide, H2O2. You know there's only one that it's going to fit into. Here's a DNA polymerase enzyme that actually helps in constructing DNA molecules. You can see that the shape at the active sites are different, so this wouldn't fit. But we look here, we know, we've even talked about this in class, that catalase, remember ASE always tells you have an enzyme. That catalase always breaks down hydrogen peroxide into oxygen and H2O, and you'll see that the active sites, they do fit, so very specific. Okay, 4, the reactions. Remember, there are going to be chemical reactions that are taking place. They're going to be of two kinds, and the first one is anabolic. Now, I always tell class that, you know, if you think anabolic, you might think steroids. Like somebody who wants to get big and wants to get big fast, they'll inject steroids, and that helps them build mass, add to their mass. And anabolic reactions, that'll help make it, if you understand what anabolic is, it'll help you understand what type of reaction this is. It's going to be one that's actually building up a molecule, and these tend to require not as much energy. So we're going to look at this picture right here. And so you can see here's the enzyme, here's the substrates. It's always going to take a little energy to get the reaction going, and we'll talk about that here a little bit later. And then once the reaction's over, they're together, and then the new compound is formed and it's over. And so anabolic, if you look, there's a bunch of smaller molecules. If you give it just a little of energy, it will form larger molecules. So it's building up a structure, like anabolic, like you would build muscle. Catabolic reactions. Catabolic are just the opposite. And so like we talked about up here with the catalase and the H2O2, the hydrogen peroxide, that is going to be a catabolic reaction because the enzyme catalase is going to take hydrogen peroxide and needs a little more energy to do it, but once it gets that energy, it's going to break it apart, and then you're going to have two new compounds. Okay, so catabolic, think about catabolic, just the opposite of anabolic. Instead of building up, you're breaking it down. So it's breaking molecules apart. It requires a little more energy to do that, but it also helps release some energy. And we'll talk about that a little bit later. And if you check out the picture down here, you can see you got this bigger molecule, and it's going to be broken up by a catabolic reaction of an enzyme, and in the process, it's going to release a little energy. Okay? Now, all biochemical reactions where enzymes are needed, they're always going to need a little energy to start. Little energy, and this is called activation energy. Usually not a lot, but you got to remember the chemical reactions that are going on in our body through enzymes. If it wasn't for enzymes, some of these would not occur in nature for millions, maybe trillions of years. And so we need these, this activation energy to start this chemical reaction of a lot of these enzymes, and so that way we can continue with life. I always like to think of it as an analogy. Like if you wanted to burn a pile of sticks or a pile of leaves, you know, if you just left them, all the energy would go back into the ground, but it would take years to do it. But if you wanted to add a little energy, you could start them on fire, get that chemical reaction going, and within, you know, a few minutes, all those nutrients are going to be able to leach into the ground much quicker. And so activation energy, enzymes lower that. And so we don't have to, you know, we don't need to get a big torch to do this. We could just use a little match, and that's just a little bit of energy. Another way you could think about it, you know, you could take a torch and you might be able to burn a pile of wet wood, but you could take a match and maybe start a pile of, at a lower temperature, you may be able to get that reaction to take place. And so the enzymes always lower the activation energy. Kind of a scary thought when we talk about if my hand is the enzyme and my fist is the substrate, they absolutely have to fit perfectly, right? Remember, one enzyme has one substrate and it'll produce what's called an induced fit. This process is random because if this substrate doesn't hit that enzyme perfectly in the active site, it won't engage it. So it hits it anywhere around, it's not going to engage the enzyme. And so it's kind of a weird feeling to know that our life continues on because of these massive amounts of random collisions that hit just right and allow life to continue.